what we're up to today is we are going to be talking about early deterministic radiation effects, and we'll have a couple of activities, and that's kind of the name of the game for us. But hopefully by the end of the day today, we'll have a really clear picture of how we do modeling for different types of dose responses, as well as a pretty in-depth look at acute radiation syndromes. And our reading has some good depictions of that and discussion of that, so we'll be building on that. Key terms did as well, so I'll be posting those this evening. But yeah, that's the big takeaway is coming up with a philosophy for largely for LD 5030. And one thing I want to stress, where we looked at the biological responses last week, the LD 5030 for cell populations is pretty much the same LD 5030 for human populations. So because of what radiation does at the cellular level, we can talk about LD 5030s for cell populations. We can also talk about them for human populations, which is typically the way that we think about them, but they're very closely related. So we will be looking at some pretty grim and grisly stuff today. And for the most part, with a few exceptions, these are not things that we will be exposing patients to or potentially be exposed to in our work in diagnostic imaging or radiation therapy. There is maybe one syndrome that we could possibly run into in the radiation therapy world, but we're not generally going to see some of the more severe radiation syndromes. So I don't want you to walk away from this lecture thinking that, oh my goodness, what have I done? Like this crazy job or whatever, like people be dying and stuff, yo. It's not like that. It's what we're doing is practicing this stuff at a very, very low level. And so in general, we are not going to see these responses. So then the general student question is, well, why the heck are we learning about it? Well, it is the foundation for what we know about radiation interactions comes from looking at large human populations like what we talked about with Hiroshima and Nagasaki. And those reports from that research data had to be expressed initially as early radiation effects. So that's why we have these early deterministic effects. They were the first documentable radiation effects that we found for large human populations. The late and the stochastic effects, those evolved later, right? Like as the name implies. So this was kind of the gateway that allowed us to see, yeah, this stuff is causing severe problems. We need to continue to monitor it. So we are going to list and describe various early effects of radiation, these syndromes, right? And we will chart what each one of these syndromes, what doses cause these responses, like kind of dose ranges, as well as what those responses are. We'll talk about LD 5030 again and LD 5060, and as well as some organ dose limits. And we'll talk about why LD 5060 is probably more accurate in terms of understanding long-term how to respond to acute radiation illnesses. We'll chart dose-response relationships for skin, blood, chromosomes, and germ cells. That's a little bit repeat of some of what we looked at last week, but it's important, and it's helpful for us to look at those organ doses, because we do, even though we're not going to be doing whole body doses like what we're seeing for these acute radiation syndromes, we do occasionally in CT, PET CT, and radiation therapy cause organ doses that can cause problems. So we do need to look at that at the organ level. So early effects are those, they're going to happen relatively soon after humans receive high doses of ionizing radiation. We're normally talking about hours, days, or weeks after an exposure. Occasionally it could be a month or so out from an exposure, but generally hours, days, and weeks after an exposure. the radiation exposure would have to be very, 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 very high to cause an immediate response within minutes. It would be so high that there would be no hope of living a few hours after that exposure. And there's not a lot of cases of that, but we will talk about one case of that. A lot of this comes from laboratory animal studies. So if you watched the Chernobyl video, the white rats and the mice studies, we've used a lot of what biologists call mice models, right? And that's a really nice, sweet way to talk about the fact that we've irradiated a lot of mice. Now, why did we irradiate all of these mice? Well, in terms of genetics, mice are very close in their genetic makeup to human beings. And they reproduce much more readily. They have a shorter lifespan than human beings. So in terms of seeing what radiation could potentially do to human populations and potential hereditary effects, a lot of those experiments have been done on mice. There is some data from irradiated human populations, and a lot of the data that comes from irradiated human populations has a big kind of ethical price tag on it. Like I've mentioned, some of this data comes from survivors of nuclear weapons attacks. Some of these data comes from World Health Organization and their studies around Chernobyl. Frankly, some of this data comes from Nazis who were doing experiments on Jewish people with using radiation. So that's what I mean by there's a huge ethical question mark over it, and there's clear and present reasons why we don't continue these kinds of experiments. So after World War II, you have the Nuremberg Trials, where Nazi scientists were put on trial and found guilty of crimes against humanity for their experiments, and some of those experiments were done with radiation. We kept the data, though, and so we continue to use those scientists' findings and their data, but we're not going to, there's no chance we're ever going to repeat any of those experiments at any point in time. So some of it is still very theoretical. And again, I want to stress it over and over and over again. These are not common in diagnostic imaging of any kind, and these are not even common in radiation therapy. These are the extreme cases. Let me move this slide around a little bit. This comes from the text that we read, but the factors that are going to possibly cause damage both to somatic and genetic cells, so germ cells, is the amount or quantity of ionizing radiation to which the subject is exposed. Now, when I talk about quantity as an X-ray tech or as a therapist, I'm thinking about the amount of photon flux. All right, I'll say that again. Quantity is something measurable as photon flux. It is controlled largely by mass or by monitor units for radiation therapists. And it is purely the amount of ionizing events that are occurring. That's the quantity. This does not translate as readily to nuclear medicine applications because the quantity in that scenario will depend somewhat on both the type of exposure that a person's being exposed to, whether it's just gamma or emitting some kind of particulate radiation, as well as its half-life. So how volatile is it? How reactive is it? And also, is it a radioisotope that's going to hang out in the system and be brought up and either stored in bone or stored in some other organ system, or is it something that's going to be readily excreted out of the body? So there's a much more stickier can of worms around radiopharmaceuticals when it comes to quantity. So we will look before the end of this class, not today, but before the end of the trimester, we will look at how we have to quantify internal doses from radioisotopes, but just recognize that largely when I'm talking about quantity, I'm talking about photon flux. Then the ability of the ionizing radiation to cause ionization of human tissues, and we were able to quantify that. Do y'all remember what unit or what expression are we using to quantify the ability of ionizing radiation to cause damage to human tissue? What unit or what concept are we using to talk about the ability of ionizing radiation to cause damage? RBE, good. And RBE is based largely on linear energy transfer, right? So either one of those terms is acceptable in a way for us to talk about its ability to do work in biological material. Then the area of the body exposed is very significant, particularly if it's an area of radiosensitive organs. So a radiation exposure to the head is very different from radiation exposure to the pelvis area, right? We would expect to see very, very different responses in those two areas. In fact, one experiment that the Nazis did, and one of the reasons that we know that the dose required to sterilize both male and females is 6 gray to the pelvis, is they would have people who were suspected of being communists or homosexual or Jewish in some of these nations that they occupied, like Poland. They would say, it's okay if you're a member of that population, come in and fill out a form. So you would sit down on a bench and fill out the form, and then the next day you would have burns in the areas of your pelvis. What they had done is within that bench there was radioisotopes that they had calculated that the time it would take you to fill out this form, you would have received sufficient dose to sterilize you. So basically you would have a serious burn and you'd be unable to reproduce, right? That was Nazi experiments that allowed us to realize that. So I say that horrible example, number one, to again emphasize that there's ethical price tags to our understanding about this stuff, and then also to emphasize that the amount or the area of the body is very, very significant to us. As well as the size of the body that's being exposed. So we have a dose area product where we look at the amount of exposure over an area, the surface area of the body that's received that exposure. And so the surface area size is significant. All the data that we have about acute radiation syndrome relates to whole body exposures, right? And the only modality in the healthcare system that deals with whole body exposures is nuclear medicine. Even in radiation therapy, I've participated in maybe one whole body exposure using a linear accelerator, and it was something that we monitored very closely. But X-ray and generally radiation therapy, we are targeting a specific area for just these reasons. So, somatic effects. And these are generally the way that we will frame our understanding of early deterministic effects of radiation exposure, is we will talk about somatic effects. Now, what does somatic mean? What does that term refer to? Cells of the body. So cells of the body that are non-reproductive cells. So these are cells that are not responsible for hereditary information or passing on new generation. They're basically not sex cells. So soma, the Greek term means body. So another way to just think about it is these are the cells of the body, right? So we're talking about the effects of radiation on any cell that makes up the organism apart from germ or sex cells. So biological damage sustained by living organisms after exposure to ionizing radiation might be another way of thinking about these somatic effects. They are generally classified as either early somatic effects or late somatic effects. And I want to further kind of classify here, and this is a part that we're going to need to unpack a little bit. When it comes to the early somatic effects, they are all deterministic in nature. They are all deterministic. That means that they have some threshold, and after that threshold, the amount of dose is directly related to the severity of the response. So the dose determines the response. The late somatic effects can generally be branched into two categories. So when we get to late somatic effects, we're going to need to put on our thinking caps a little bit and figure out, okay, is this a deterministic effect or is this something else? And so they could potentially be deterministic, and I'll make that little sign there just for ditto. They can also be late deterministic effects, or they could be stochastic. Does anyone remember what I've said that stochastic means?
No threshold.
So there's no threshold for these. What else?
All radiation is safe.
It does lead us to conclude that all radiation is not safe. But stochastic largely means that it is random in nature. So it's not the severity of the response that is determined by the dose. It is the likelihood of the response. So the likelihood of a stochastic response increases as dose increases. The classic example for stochastic effects is cancer for radiation. That as our work as X-ray techs, as we work longer and longer in this field, we run that much more of a risk of someday developing cancer. That's the truth, right? That's the truth for every single one of us in this room, right? So being aware of our annual dose limits, practicing radiation safety, especially if we're dealing with radioisotopes, all of those things will help minimize that dose and therefore also minimize the likelihood of any long-term problems. So, the reason I'm spending a lot of time on these kind of boring slides is that a lot of students get confused about the early deterministic effects, or early, like if we were to talk about early somatic deterministic effects, all of those words just start to blur together a little bit. Each one of these words is very scientifically precise in its use, and so our reading, one of the things I like about our reading is that it does try to parse out as carefully as possible. Like for the rest of this lecture, I'm pretty much just talking about early deterministic effects, right? And they are going to be early deterministic somatic effects even. There's really only one slide moving forward that deals with heredity. So to break that down, I'm talking about things that happen within hours or maybe weeks after exposure. That would be the early part. They are determined by the dose. So as the dose increases, the severity increases. That's the deterministic part. And the somatic part is within the cells of the body, right? Within the soma or the cells of the body. So, as if that were not confusing enough, there is old documents that are out there that refer to deterministic effects as non-stochastic somatic effects. That's the way that I memorized it when I was in school. Non-stochastic just means it's not random. It's not random. So deterministic is a more precise term because it's not saying it's just not something. It's saying it's determined by the dose. So here again is the most concise way to put that as radiation doses increase, the severity of these effects within the body also increase. These results have a threshold or a point at which they begin to appear and below which they are absent. So that's good news, right? That for us, if I eat a banana, I'm not going to hit some threshold and my hand fall off, right? there is a threshold for every single one of these dose amounts. And after that threshold, that's when we start to see the severity increase. So the way that we typically express that with a graph is we would have some kind of response over here on the y-axis. So this is where I would illustrate my response. And over here I would have a dose, and the amount would be increasing as it moves in that direction. And the responses would also be increasing. Oh. Let me move this. Everyone tracking with me so far? Now, for these early deterministic early deterministic somatic effects, which model do we probably need to choose for a response? We've said that the response increases with dose and it has a threshold. So how are we going to express that graphically? Any guesses? It is like a deterministic graph, yeah. It has a threshold. So let's go ahead and write here's a threshold right here. So we say right at this point, at this dose amount, whatever that dose amount is, we start to see a response. Does anyone know what shape we should be drawing on here? It's the shoulder thingy. That's right. So good. It's going to have an S shape. It is what we call nonlinear. Now you'll say, well, that still looks like a line to me. But the point is it's not a straight line. It's a curvy line. It looks very similar to the H&D or characteristic curve. So let me try to draw it one more time. Okay? And so remember we talked about how it has a toe, it has a slope, and it has a shoulder portion to it. Right? Probably the only thing that we could potentially be exposed to in our work in medical imaging as it stands right now and therapy, but there are some people who work like in the cath lab who receive a high amount of radiation exposure to the lens of their eyes. There is a threshold of 1 gray cumulative over the course of your lifetime that you could eventually get cataracts. The likelihood of cataracts increases from that point on. Now, you can imagine that has become a highly controversial thing, and so there's a lot of debate in the literature about how accurate that is. That is something that we could design an experiment around without further jeopardizing people, because there's a lot of people who work in cath lab, and we could figure out cumulatively what that means. The reality is it's going to be pretty different for every person, and that data would require a long time to record. It'd be a very complex study. So the point of the matter is, if you work in cath lab, get yourself some dang lead glasses, right? And be the nerd that's wearing lead glasses. People can make fun of you, but then you can make fun of them the day after their cataract surgery, right? So that is the only potential hazard that I've seen related to our work. And that would not be an early deterministic response. That would be a late deterministic response. It would take years. So we will look at it a little bit more next week. But here again is that model. Let me move this up just a little bit. This is found in the text that we read, figure 8.1. It is a threshold nonlinear dose-response model, and this is the model that we use to talk about these early deterministic somatic effects of radiation exposure. So there will be some kind of threshold. If we're talking about people dying, right, populations of people dying, this chart shows us just that there's a point, a threshold, and for us the threshold's about 1 gray again. And at 1 gray, we do start to see people die. and this is a whole body dose. Their whole body is exposed to 1 gray of radiation from any source. We can try to mathematically map that all out and get an effective dose. But I'm just talking about 1 gray of exposure in air or in tissue. We would have hit the threshold to start to potentially, if someone's immune compromised or aged or a weaker part of the population, they could potentially die after that 1 gray of exposure. Now you're going to start to see it ramp up significantly around 3 to 4 gray. So this midpoint here, where this is significant because graphically this midpoint is the LD 5030, right? LD 5030. So at this point, and we'll look at this a little bit more, but at this point, 50% of the population will die within 30 days. The other 50% may live longer than 30 days, but they will likely die as well. So it's not as though they go on to have a happy life. No, pretty much everyone dies at this point. Just some people live past 30 days. And if you ask me, I probably would, at that point, want to be one of the people who's in the 30-day club than the 60-day club, because it's not a lot of fun, those extra days. This is 3 to 4 gray, whole body dose. Far and away, thousands of times higher than any radiation exposure that we give in any of our modalities. Okay? This slope has a shoulder on it, right? Because once you get right here, guess what? There's no one left to kill. And so we call that the overkill point. Any radiation received beyond that is just gratuitous. So we will look at some cases of people who received radiation exposure sufficient to have killed them 50 times over or whatever, and how that played out. So these early deterministic effects, again, appear within minutes, hours, days, or weeks from the time of radiation exposure. It requires a substantial dose of radiation. There are only really 2 potential modalities that could cause this. They would be radiation therapy and interventional radiography. And when I say potentially could cause this within a person's working day, those machines and those modalities have contributed to early deterministic effects. There are documented cases of, generally through error or through, frankly, stupidity, someone accidentally killing themselves. For example, when I worked in Utah in the Salt Lake Valley, there are more linear accelerators than anywhere else on the earth. They use them for cancer treatment. They also use them for a lot of military applications, like they basically build CT scanners that have linear accelerators instead of X-ray tubes inside of them. And then they use those gigantic linear accelerator CT scanners to CT scan rockets and airplanes and missiles and things like that. They do that to inspect the parts for damage in construction. So who here remembers the Challenger explosion? Was anyone alive when the Challenger spacecraft blew up? 1982? No. I think I was in 2nd or 3rd grade. They stopped class. We all sat down. Everyone was excited. There was a space shuttle launch, and they'd build it. It was, I think, the first female astronaut was on the shuttle, and it got about halfway into orbit, and the engine blew up. All the astronauts died in an instant. It was a big deal when it happened. The company that constructed those engines, Thiokol, was based in the Salt Lake Valley, and so people were building, Boeing and people who, Lockheed Martin, who build missiles, use these linear accelerators to try to prevent that kind of thing from happening, right? So one day, these engineers are working on a rocket at Phi Call, and they decide to prop the vault door open. So they tape over the safety latch on the vault so that they can leave the vault door open and run the linear accelerator at the same time, right? Because they've got a deadline and they're trying to get this engine out, and they're having to run this giant linear accelerator, and the extra 30 seconds or whatever it takes to close the vault door, they don't want to waste, right? So they're running a linear accelerator with the door open, which you can do. You can tape the vault doors closed, right? Or trick it. It's just like the light in the refrigerator. If you tape the light in the refrigerator, you open the door open and it still thinks the door's closed, right? These guys, one of them died of acute radiation syndrome from operating a linear accelerator. So, and I'll tell you too, as a radiation therapist, I have also taped the door device down. Why did I do it? Well, it was like our last treatment of the day in the service. We'd already called the service guy. We weren't going to be able to do this person's treatment unless we basically fooled the machine into thinking that the interlock was clear. Did it make us nervous? Yeah, it did. Did we document it? No, we didn't. So basically I'm saying don't be a dummy like me. Or if you are going to do something like that, be aware of the hazards that come with it. So whole body doses of 6 gray can result in nausea and fatigue, erythema, epilation, blood and intestinal disorders, fever, dry and moist disquamination of the skin, and temporary or permanent sterility in male and females. And we'll just go ahead and assume it's going to be permanent sterility at that dose amount. Here is an example of the types of erythema. This man is an early responder to Chernobyl who received severe radiation burns to his arm. So let's talk about what these acute radiation syndromes are, how to identify them, what are the stages of the response, etc. So the first thing for us to think about are these stages. So all patients, regardless of the syndrome or the body response that they experience, have the same stages. The only thing that changes may be the length of the time of some of these stages. So it is important to know the names of each one of these stages. Okay? They are prodromal, which is the very first stage. And this is an immediate response. So a person's acutely exposed to radiation in excess of 1 gray, and they may experience nausea, vomiting, and diarrhea. The severity of that prodromal response and the quickness of the prodromal response vary based on the dose amount. So if they just received 1 gray, the severity of the nausea and vomiting will be less than if they received 10 gray. It will be significantly worse. During a latent period, something very strange happens. It seems that nothing is going on. The person may still feel a little queasy, but otherwise they feel healthy, they feel active, they can go about their business like normal. Then they enter into a manifest illness. And this is when we see the specific syndromes that we've identified. The 3 syndromes are the manifest illness. One mnemonic that Tom Wolfe has proposed for remembering these stages is please let me die or recover, right? Prodromal, latent, manifest, and then you either die or you recover, right? So PLM, please let me die or recover. That may be the way to remember these stages of response. So let's look at kind of an overview of how these stages map onto different dose amounts. So we've said with 1 gray, we could see any of the nausea, vomiting, diarrhea, fatigue, leukopenia, which is just decreased white blood cell count, or nothing at all. The person may, there are some people who are fairly radio resistant as individuals, and they do better. In fact, women as a whole, when it comes to whole body doses, women are more radio resistant than men. Why that is, no one knows. That latent period, once we, in doses in excess of 1 gray, we will see a latent period where nothing happens. And that latent period, like I said, will last different lengths of time based on the amount of dose. So, like for hemopoietic, the latent period can be upwards of 2 weeks. For CNS syndrome, the latent period is generally a matter of hours. That hematopoietic syndrome is at doses of 1 to 10 gray. The average survival time is just over 30 days. And the manifest illness has in particular the diarrhea, the decreased white and red blood cells, hemorrhage, GI infection. Infection. So it is largely the people die due to either infection caused by the decreased white blood cell counts or just anemia. Any little cut that they get causes them to bleed all over the place, and the bleeding never stops. GI syndrome, so around 6 gray, and you see there's some overlap between these different syndromes. 6 gray and above, the average survival time is a matter of days, so 3 to 10 days. And during the manifest illness, we'll see a severe nausea, vomiting, diarrhea, fever, lethargy, and then generally what kills people off on this is either the electrolytic imbalance or emaciation. What's happened is there's been a complete denuding of all the intestinal cells, and so the intestines have lost their ability to extract nutrients from foods. Finally, the CNS syndrome is doses at 50 gray or higher, and this is the maximum survival time for this is 3 days. And it is, you have the same prodromal effects, but the latent period is much shorter. And ultimately, you have all of this fun stuff you get to experience of excessive nervousness, confusion, loss of vision, loss of consciousness, shock, agitation, loss of equilibrium. Plus, generally, your heart just stops working. Your brain stops telling your heart to beat. In fact, you will die sooner than having like the diarrhea or anything like that. So you would not become anemic or anything. Your body would not have sufficient time. So here is yet another way to think about that or look at that. And we said that 1 rad equals 1 centigrade, right? So 100 rads equals, again, what? 1 gray. There's that number again, just in a different unit of measure. And it gives you the rough amounts of time related to these stages. So if we are responding to an acute radiation situation or some kind of terrorist attack or something that's requiring us to respond as healthcare providers to this, we need to be able to figure out, calculate what was the dose amount that they received And also we have to record what are the responses that we're seeing and what's the time frame after exposure that we're seeing these responses. So here is a graphical representation of that same response, where we see over time the symptoms increase over the prodromal period. During the latent period there may be a slight fall off. The person may be able to return to normal. And then the manifest illness, the responses increase again until that point of the shoulder. So let's look at each one of these syndromes. The first we'll look at is hematopoietic. These are doses between 1 and 10 gray. The prodromal stage will begin within hours after exposure and persist for days to weeks. This pancytopenia just means that throughout the entire blood system, white blood cells, T cells, red blood cells, all of them experience decreased populations, and that results in either infection or hemorrhage. Death after 2 gray is in 6 to 8 weeks for sensitive individuals. 4 to 6 gray is the range of the LD50-30. I've said that the LD50-30 for most human populations is 3 to 4 gray, so I'm going to stick with that. But you can see there's pretty big scientific debate about what exactly is the LD50-30. The LD50-60 is kind of loosely what I'm basing that 3 to 4 gray off of, so we'll look at that in a minute. After 10 gray, all die within 2 weeks unless given bone marrow transplant, and that has rarely proven successful. Okay, the GI syndrome is for doses between 10 and 100 gray, more accurately probably from 6 to somewhere around 25 gray. But at doses in these range, we would expect to see some kind of GI syndrome if the person lives long enough. Death is independent of dose. All die at roughly the same time, and that's 3 to 10 days without medical intervention or 2 weeks with medical intervention. And it's largely the result of intestinal denuding. The central nervous system syndrome occurs at doses over 50 gray. The latent period ends after 5 to 6 hours, and death occurs in 2 to 3 days. They will experience nervousness and confusion. The cause of death is not fully understood, and the autopsies will reveal very little cellular damage because they have not been alive long enough for the manifest illness related to the blood systems or the GI systems to show all of the DNA damage and all the cell damage that's occurred. Probably the most famous case of this was a KGB double agent. I think his name was Alexander Lepetsky. I can't remember how to say his last name. But he was poisoned with polonium by KGB agents. They put the radioisotope in a cup of tea, and he drank the tea. And I think I've shared this with you. It was the most dangerous autopsy, the most dangerous post-mortem ever done in the history of the world. They had to impound his car and his condo and pretty much all of his personal effects. He died of the central nervous system syndrome within 3 days. Actually, I think it was within 2 days. And his heart just stopped working. So, again, the cause of death is highly debatable, and it's inevitable, though, at the same time. So let's talk about these LD qualifications that we have. The LD50-30 signifies the whole body dose of radiation that can be lethal to 50% of the exposed population within 30 days. It's a quantitative measure that's fairly precise when applied to experimental animals. So again, when we use the mice populations, we can get into a ballpark range of what this would be for human beings as well. The LD50 for humans may require longer than 30 days for its full expression. For adult humans, it is estimated to be 3 to 4 gray. If you're looking for a number to memorize, that's it for the LD50-30. So, and I think in the key terms also it asked, what is the LD50-30? That is the numeric LD50-30 for human populations. Here again is that graph, on this particular graph, they've kind of split the difference. And they've given us our threshold at 1 gray where we start to see a response. But the 50% line you can see is close to 3.5. So they kind of split the difference between 3 and 4 gray. And then I guess the further thing is to note where the shoulder is for the overkill point. They've marked as 6 gray. So in excess of 6 gray, we do not expect to see people recover. Now, there are different ways that we can talk about radiation dose and responses. So we can have LD50-30, 50-60, and 160. Each one of those represents both the percentage and the time. So the LD10-30, 10% of the population in 30 days. LD50-60, 50% of the population in 60 days. And LD160, 100% of the population within 2 months. What would be the dose required to kill everyone? All of these measures refer to the percentages of subjects who die after exposure. And I want to stress that this is the way that we quantify and the way that we think about acute radiation syndromes. But there's LD50-30s for everything. Like there's LD50-30s for water. Like how much water could someone be exposed to before they die within 60 days, right? There's LD50-30s. I was making popcorn this afternoon for an event here. They probably have to publish LD50-30s for popcorn or the materials that are used inside the popcorn, right? So this is also one of the reasons they have to post warnings on gasoline pumps. There are LD50-30s for exposure to gasoline fumes, just the fumes coming off the gasoline. If you sat there and breathed gasoline fumes long enough, eventually a percentage of the population would die, right? So we use this, I guess what I'm stressing is that these ways of quantifying things are not solely the domain of radiation biology. They're used by biologists to quantify all exposures to all sorts of things, mostly chemical things. So why do we have these different amounts? Well, the LD50-30, the LD10-30 gives us a minimum or a baseline. That's where that 1 gray comes from, right? So it gave me the threshold amount where I start to see a response, right? These LD50-60s and LD160s, that is the overkill portion of the curve, right? So if I put that, these amounts are all important. They show me the shape of that curve, right? So the 1030 would be like a baseline or the threshold at which we expect to start to see responses, and the 160 would be like the overkill portion. The LD50-60 may be a little bit more accurate for humans because humans, we have the ability to medically respond to the damage. So we can do blood transfusions, we can do bone marrow transplants, we can give people fluids if they have an electrolyte imbalance, we can give them medication that can help them not go into shock. So it may be more accurate if we're talking about for a person in an exposed population who receives medical treatment, it's probably better to look at the 60-day window for survival. And of course, humans are fairly complex organisms. So within our cells, we have the capacity for repair and recovery. We've already talked about repair enzymes and how they work to repair damage to the DNA, as well as damage to the cell itself. So after irradiation, surviving cell populations begin to repopulate. And the amount of functional damage sustained determines the organ's potential for recovery. So there will probably be some kind of scarring or some kind of permanent damage to organ systems. But as long as the scarring or the damage is not completely disabled the organ from functioning, if the cell populations are able to recover, the organ will be able to come back online. Now, it's an interesting thing. We've talked about how there's an oxygen enhancement ratio for biological damage that's caused by ionizing radiation. But there's also an oxygen enhancement ratio for repair. So once the damage is done, it is good for there to be more oxygen. These findings led people in the therapy realm to look at questions related to if we put people in hyperbaric chambers or give them oxygen-rich environments after radiation therapy treatments, will there be enhanced cellular repair? And largely that research has been inconclusive. But this is the case for these acute radiation syndromes. Repeated radiation injuries do have a cumulative effect, and that is largely coming out of what we found about cataracts. And approximately 10% of radiation damage is irreparable, right? And there may be upwards of 90% of repair over time. So, moving out to the local tissue level, moving out from the cellular level, a destructive response in biological tissue can occur after high radiation doses. This leads to atrophy of the organs and disease. So everything that we've highlighted in terms of the radiation syndromes and how those cell populations were impacted, so decreased of white and red blood cell counts, decrease in intestinal lining cells. All of those decreases have a mirror effect at the organ level. And so if, for example, we have a person's liver, which is a fairly radio-resistant organ, but if it's exposed to a large amount of radiation, there will be damage within those liver cells, but the organ as a whole will continue functioning, and it will go through repair processes. But there will still be some kind of atrophy or some kind of weakening of that liver. The skin effects are pronounced with radiation exposures, especially to gamma or X-ray radiation exposures. So this is a woman who has severe skin burns after exposure to radiation from the nuclear bombs. It's actually burned the pattern of her kimono onto her back. The term for that is radiodermatitis, and specifically desquamination. And there are radiation therapy patients who experience this. The skin will appear red and generally moist because the cells have stopped working to retain moisture, and so the skin it is not sweat, it is moisture that the skin cells are incapable of retaining within the body. Radiodermatitis, if we do this for prolonged periods of time, so those early radiation X-ray techs who had the hands who were so severely scarred. The scarring of their hands caused loss of capillaries, so loss of blood vessels within the hands. The loss of blood vessels resulted in necrosis, and the necrosis resulted in amputations. So we can have radiodermatitis that either results in cancerous lesions or other severe impacts. Another example is epilation or loss of hair. So moderate doses of radiation may cause temporary hair loss, but with larger doses of radiation, the hair loss may be permanent and irreversible. And I always think about folks who are undergoing radiation therapy for whole brain treatments. We generally get loss of hair just in the area that we're treating. And it's a very clear pattern of the treatment portal where the hair is lost. And so we would typically tell people to go ahead and shave their head prior to starting treatments. Hematologic, I gave us this number last time, and it's a pretty hard and fast number. So whole body doses of ionizing radiation, and I would even say partial body doses of ionizing radiation as low as 0.25 gray can produce a measurable hematologic depression. So that is CT, radiation therapy, and PET CT, as well as potentially some fluoro examinations can cause some amount of hematologic depression. And the potential side or the potential effects, health effects from that are infection and anemia. So let's talk a little bit about how this system works, and this is going to help us too so again, we're still talking about somatic cells of the body, but thinking about how this depression cycle works is helpful. So we have these stem cells here at the center, or they're sometimes called pluripotent cells. They have the ability to go in one of these four different directions. They can become thrombocytes or platelets, they can become lymphocytes, they can mature into granulocytes or erythrocytes, red blood cells. So these pluripotent cells, or these stem cells, have all the coding to be any one of those other cells. Since they are what we would call undifferentiated, they have not yet differentiated into a red blood cell or a granulocyte. Since they are undifferentiated, they are the most radiosensitive cell in the body. So the stem cells of our blood system are the most radiosensitive cells in the body. That's why we can say that the 0.25 gray can cause hematologic depression. It's not going to happen overnight, though. What's going to happen is a dose of that amount is sufficient to kill a lot of the stem cells, right? But stem cells are not circulating around in the blood. They are within the bone marrow of our long bones, right? So the stem cells are within the bone marrow of our long bones. What's circulating around in the blood is mature blood cells, so erythrocytes, granulocytes, lymphocytes, thrombocytes. That's what's circulating. Since they are differentiated, right? Since they are mature and differentiated, they are more radioresistant. So they are not going to die. The same dose that killed all the stem cells in this model did not kill the red blood cells. So what's the problem? Well, the problem is that 2 weeks from now, when the red blood cells start to naturally die off, there is not a next generation ready to replace them. There's going to be a hang time there where the body has needed to reproduce more stem cells. And during that hang time, that's when you have the erythema, the anemia, the leukopenia and all those decreased cell populations. Not because we killed off the adults, because we killed off the kids, right? So there's no kids coming up to replace the cell population at the appropriate time. So that again is why there is this prodromal period, why there's this period of latent illness, I should say why there's this period of latent illness where no effects, no observable effects are occurring, because there's not sufficient, the time is required for those mature cells to die off. So when we are looking at these cytogenic effects, we look quite a bit at chromosomes because we're able to see under a microscope during metaphase, we can see how the radiation has destroyed the DNA. So it is not just, it's wiped out the cell population and made it unable to reproduce in a way that the body didn't immediately recognize. Sometimes we call this cytogenic analysis a karyotype. And it's like a photograph taken during metaphase. So metaphase is the phase of the cell cycle when we can document, we can view chromosome damage. During any other phase of the cell cycle, if we stop the cells and investigate them with a microscope, we will not be able to see the damage. It is only once we have those chromosome bundles during metaphase that we can observe. Here are some karyotypes showing us what these different types of damages look like. And so if you see the top row, these are probably normal. But as we go down, like to row F and G, these are fragments and pieces of chromosome that have been broken off due to radiation responses. Okay, last slide here. So I do want to, since we spent most of the time talking about somatic effects of these early radiation things, and that's generally the way that we're talking about, because we're not talking about people surviving past 60 days when we talk about this stuff. So there's no way for them to reproduce themselves and go on about their life. But there are impacts on the genetic cells or the germ cells as well, these sex cells. And it is very similar to what I was talking about with the blood cell populations. So we've got these primordial cells, or these pluripotent type cells that have not yet differentiated. So the spermatogonia and this primordial follicle are fairly undifferentiated, therefore they are more radiosensitive. By the time you get to the mature ovum and the sperm cell, those are highly differentiated cells, and so they are not as radiosensitive. They are more radioresistant. But if there's a sufficient radiation dose to kill off or depress the undifferentiated cell populations, the cell population of the spermatogonia, then after a period of time, after a period of weeks or days, eventually that impact will be seen with like sperm counts, right? And it's just the exact same model of what Burgony and Tribando told us about. Differentiated cells are going to be more radio resistant than undifferentiated cells.